My name is Doroteya, and I'm part of a UK-based data analytics company called Multicore Dynamics. And I would like to share one of the methods that I'm using to integrate omics with non-omics data called Topological Data Analysis, or shortly TDA. This method is rooted in algebraic topology, a very old branch of mathematics dating back in the 1700s, when mathematicians were trying to describe properties of objects that are preserved under continuous deformation, like the one shown here in the middle. So albeit coffee cup and a donut are geometrically different objects, they're topologically equivalent and can be described with a single hole. On the other hand, a spoon which has no holes or teapot which has two holes are topologically different objects. Why this is important? With advancement of computing technologies, new breed of tools that are trying to capture and explore shape and connectivity in data emerged. They're, of course, rooted in algebraic topology and are called topological data analysis. And quickly become apparent that these tools are very powerful in filtering noise and revealing structure in data that is invisible in mainstream statistical tools. The main algorithm used in TDA is called mapper, and as the name suggests, it refers to constructing a map. So it is starts with assuming that input dataset is sampled from a point cloud, as the one shown here on the left, and then a mathematical operation can be applied to it. For example, a distance of each point to the most left located point can be estimated and used to construct one-dimensional topological lens or filter function with values ranging between min and max, as indicated with red and blue here. So then the range of values is beamed with overlapping beams, and clustering is performed whereby each cluster of samples form a node, and two nodes are connected with an edge if they share one or more samples. So in this way, a graph representation of the shape of data is obtained as seen through the one-dimensional lens. And this representation aims to preserve the connectivity and sample proximity from original data space. So the mapper, of course, works with also two-dimensional lenses when the values of the filter functions are used to construct overlapping rectangulars where the clustering is performed. And of course, the output is again a graph representing the shape of the original data cloud. As you're all aware, understanding complex biological systems requires collection of diverse data modalities that can provide a complementary view, and that comes with the challenge to integrate or connect omics with non-omics data, or to infer insight across these diverse modalities. So in this line of thought, TDA can be of help because each of the data modality can be encoded as topological lens and then fused together in a unified topological network, which carries the information about connectivity and shape of the biological samples across different data modalities. How this is done in practice, here is given an example of taxonomic and functional profiles of gut microbiome samples from healthy individuals. These data are available in open repository as published here. And the data set can be imagined as a high-dimensional manifold in which the coordinate axis span across taxa, pathways, health scores, etc. So then the location of the sample in this manifold, in each sample, can be fully determined by its taxonomic or functional profiles. So I have used this conceptual view to perform analytics from topological analytics of this data set. And the first step was to construct a topological lens, and in this case I used PATE, a non-linear dimensional reduction technique, which showed a clear branching structure in the healthy microbiome samples, with a largest branch showing a clear gradient in Shannon entropy and mid to high health scores. So then I have used the mapper to fuse the functional and taxonomic profiles as seen for the PATE lenses. And this is the topological map that was generated. Each node corresponds to a cluster of samples, and they're connected with edge if they share one or more sample. And the color corresponds to average value of Shannon entropy that was associated with the samples clustered in each node. So as expected, most of the samples clustered in a well-connected central component of the network, which indicate very similar profiles. However, it was interesting to see that a lot of samples have distinct profiles, so they were not connected with the rest of the network, but also that this appeared, the topological view showed a distinct flares or areas of the networks are shown in blue and yellow here. And the samples in these areas also exhibit significantly different health scores and Shannon entropy values. And this was further confirmed when looking at the taxonomics and functional profiles, that the samples indicated with the blue here were enriched in Prevotella copri and not much other species, whereby the group shown in yellow had diverse composition with and contains different species, microbial species. Those two groups of samples also exhibited distinctly statistically different patterns in their function as well. So to conclude, from topological point of view, healthy state is not a single homogeneous state, but rather a spectrum of states with characteristic composition and function. So I believe this is important to be taken into account when comparing with disease cohorts. Another example that I like to mention is coming from organ-on-chip models, and particularly 3D engineered heart tissues that were used to investigate the effect of the drug doxorubicin, which is known for its detrimental effect on heart. And the high-dimensional manifold was constructed using metabolomics features from three targeted metabolomic sets, contractile force, time points, and treatment dosages. In this case, I have used metabolomic sets to construct topological lenses and fuse them in the model shown here, which was able clearly to distinguish treated from not treated samples, which are also found to be associated with different patterns of force of contraction. And we use this model to infer metabolic rewiring of cardiac models when subjected to different doses of doxorubicin, and the paper is listed here on the top. The last example of complex biological system that I want to mention is coming from studies of inborn error of metabolism, particular one of the severe forms of these disorders called classical galactosemia, which is caused by deficiency in GALT enzyme, leading to toxic accumulation of galactose 1-phosphate, which can damage different organs in bodies of children. So it is paramount to quickly diagnose and treat this condition. So the high-dimensional manifold considered in this study was constructed using targeted, untargeted metabolomics, lipidomics, glycoproteomics, and free categorical clinical descriptors. And I have used each omics to construct a topological lens and then fuse them together in this unified topological model of comparing classical galactosemia with controls. And two distinct groups of patients emerged, which also showed distinct patterns across, one of them across glycoproteomics layer and the other across the lipidomics layer. So this can be further information be used to find what are the molecular drivers behind these two groups and how this can help the diagnosis and management of this condition. So if you have questions or you're curious about the shape of your data, please reach out and thank you.